Welcome to another How to Science 123 video. Today we're going to be talking about macromolecules. So before we really get into the bulk of what each macromolecule does, I kind of want to go over the four types of macromolecules. So I'm pretty sure most of you guys have at least experienced one of these macromolecules, even through things we've eaten and also just, through word of mouth. Most people have heard what each of these macromolecules are. So we have four types. We have proteins, we have carbs, or short for carbohydrates, we have nucleic acids, and we have lipids. These molecules are very large, hence the name macromolecules, and they create polymers, and their subunits are referred to as monomers. So, for to look at this image here, we can see that we have a long chain, which is referred to as a polymer, but this chain is made up of these small subunits referred to as monomers. So the more of these monomers that join up make up a polymer. So if we were to look at carbohydrates, a big example of a polymer for carbohydrates are starch. One big example for humans, starch is more important in plants, is glycogen, and it's just a linkage of these glucose molecules or these monosaccharides, which are the monomers of these carbohydrates, to one another to make a polysaccharide. Proteins, their main subunit or their main monomer is an amino acid, and the nucleic acids, their main monomer is a nucleotide. There's two types of nucleic acids, right? We have DNA, and then an important one is RNA. Lipids are very interesting in the fact that they don't have specifically monomers and they are not considered polymers. This graphic says they are not considered macromolecules. It is really just dependent on who's telling that to you, I guess. Some scientists or some people consider lipids as not macromolecules because they don't have a foundational polymer or they don't have a foundational, monomer that makes up a polymer, right? They're just the lipids. They're just there. There's no, foundational piece to them that differentiates them from nucleic acids, carbohydrates, and proteins. They're just lipids. And so that's kind of the reason as to why they aren't considered macromolecules. But if you were to ask me, I consider them macromolecules because that's what I've been told my entire life. So before we really get into what each macromolecule does, I want to go over this really important step. We have two things. We have a dehydration reaction or dehydration synthesis, and we have hydrolysis. Now you may have heard about these terms before, and the names are pretty much dead giveaways into exactly what they do. We can start off with dehydration reaction here, right? If we want to make a polymer, right, we're going to need to join all these monomers together. And how does the body do that? The body does that through a process referred to as dehydration synthesis. And the name is pretty much what it says. Dehydration, we are taking a water out to synthesize two things together. So essentially, if we were to use this example here, this short polymer has a hydrogen attached to the end of it, and then this unlinked monomer has a hydroxyl group attached to the end of it. And this makes up an H2O, right? Two H's and an O, and that's going to be taken out to join this short polymer to this unlinked monomer to make what we refer to as a longer polymer, right? So that's how these polysaccharides are made. That's how these gigantic proteins are made. Um, the DNA that we know of, the linkage of our nucleotides, for example, adenine to thymine, those are all connected through a process referred to as dehydration synthesis. Hydrolysis, on the other hand, is the complete opposite, right? We're taking a water out to link two things together. So what would be the opposite of that? The opposite of that is we're adding a water to break two things apart. So the dead giveaway here is lysis or lysis. Lysis in Latin means break or, cut essentially. And so essentially we're adding a water to cut two things away. So we have this longer polymer here, and then we're going to add water to break it back down into this short polymer and this unlinked monomer. So these are two important processes to kind of just symbolize how these macromolecules are made and how these monomers turn into polymers and make up these gigantic macromolecules. The first macromolecule I want to talk about are carbohydrates. I'll tell you right now, carbohydrates are not my favorite because to me they are a little more complicated compared to most, I guess at least for me, but we'll go over them right now. So carbohydrates include sugars and polymers of sugars. The simplest carbohydrates are monosaccharides or simple sugars, and they're usually multiples of C. For example, glucose is C6H12O6, and they are the main source of energy for the body. Glucose is the main source of energy for the body. If you think about something like cellular respiration, right, we're going to need to break down that glucose to help form ATP, and that's what makes it so very, very important. So carbohydrate molecules are polysaccharides, polymers composed of many sugar building blocks. So if we were to look at something like glycogen, which is just a linkage of glucose molecules and glucose molecules and glucose molecules, right? Glucose is considered a monosaccharide, and we're linking a bunch of those monosaccharides together to create what we refer to as a glycogen molecule. And glycogen is very important for glucose storage in our body. It's very important for if our blood sugar gets too low, right? Glucagon, a hormone, is going to require, it's going to tell the insulin, excuse me, not the insulin, it's going to tell the liver cells to break down the glycogen in our liver to release glucose into the blood. And the reason for that is because glycogen is a polysaccharide and it's linked with just these long, long chain of glucose molecules. And that's what makes it very important. Another example of a polysaccharide is starch. Starch is important for storage structures or plastids in plant cells. So it's a very important structural component for plants. So a disaccharide is formed when a dehydration reaction joins two monosaccharides together, right? We have monosaccharides, we have disaccharides, and we have polysaccharides. I'd say that monosaccharides and polysaccharides are the most foundational important ones, but if you wanted to know a few disaccharides, you can look at two glucoses linking together to create maltose, which is a main component of beer and alcohol, for example. So if you want to know why beer is so calorie dense, it's because just imagine it's double the glucose. Um, and then we have the dehydration reaction and the synthesis of sucrose, which is the linkage of glucose and fructose together. And notice how we have this dehydration reaction occurring here, right? We go back to the last slide and we refer to as the dehydration reaction being a synthesis, right? Dehydration synthesis, we are synthesizing or bringing together two molecules to create one molecule, a longer molecule. And that's exactly what's happening here. Glucose is essentially getting rid of a water to join this glucose and this other glucose molecule together to create something we call a maltose. And a maltose is just two glucose molecules linked together through a dehydration reaction. And we refer to this linkage or this connection between um, two monomers of carbohydrates as glycosidic linkages. And the same thing happens here with glucose and fructose. We see a water taken away, and now we have a glycosidic linkage between fructose and glucose, creating something known as sucrose. I want to talk about cellulose a little bit because I think it's a pretty important thing to understand. Um, there's different types of glucose. We have two types. We have alpha glucose, which is what we prefer, and then we have beta glucose, which is something that we can't actually use. And cellulose is a very big component of beta glucose. The biggest store of glucose or source of glucose in the planet is cellulose. And if you look outside your window and you see a bunch of grass and you see a bunch of trees and these leaves, they are abundant in cellulose. Cellulose is a very important component of the plant's cell wall. But if you were to take a bite out of that piece of grass, or for example, paper, which is made out of cellulose, you would notice that it's made out of pure glucose, but it is actually something that we cannot taste, right? If I were to ask you to, just bite into a piece of paper, you would taste nothing. You would taste, no sweetness. It would taste almost bland. It would taste a little, weird. And that's because cellulose is a beta glucose molecule, and our saliva enzymes can't actually break it down. So we have all this abundant source of glucose around us with cellulose, which is just literally everywhere, like trees, plants, grass, literally your front lawn, but you can't eat any of it because our saliva, or essentially our body, can't digest that beta glucose molecule. And if we were to look at an alpha glucose and a beta glucose molecule, what would be the biggest difference between each of them? It's just this, if we were to look at this one prime end of the carbon, we would just see that this hydroxyl group has been switched from the bottom to the top on the beta glucose. And that changes a lot of things because if we were to look at cellulose, which is a 1 through 4 linkage of beta glucose monomers, we would see that the glycosidic linkages would alternate from top to bottom to top. And this one single change in just flipping the the orientation of the one prime end is changing how our body can actually digest, right? It's completely disorientating our saliva enzymes, for example, amylase, from digesting this beta glucose molecule. And obviously, there are organisms on Earth, for example, cows and grazing animals, they're able to digest these macromolecules, but that's because one of their four stomachs has a certain bacteria which can break down those beta glucose molecules or beta glucose monomers in cellulose. And that's also another important fact of how we are not as evolved as some may think, right? We may be the smartest species on the planet, but we are definitely not the most evolved. We have bacterias out there that are located in the stomach of these cows and these herbivores that are able to break down this beta glucose, which is something we cannot do. And that's just a factor of bacteria and these small microorganisms being on this planet for so long and it, developing these interesting, traits and these interesting things about them over millions and millions and millions and billions of years. Next, macromolecule, we'll be talking about our lipids. They are the one class of large biological molecules that does not include true polymers. Inside the lipid, we have three types. We have fats, we have phospholipids, and we have steroids. Fats, we need it. It's an important part of our diet and insulation and a bunch of other things. Phospholipids make up our membrane. They're amphipathic, which means they're both polar and nonpolar, so they can interact with water, and they can also not interact with water. Steroids, hormones, and nonpolar. An example of a steroid is cholesterol, which is a pretty big component of our phospholipid bilayer, and certain features about that, which I'll talk about in the next video when we go over the phospholipid bilayer and the cellular membrane. The unifying feature, though, of all these lipids is that they mix poorly, if at all, with water. Some lipids, for example, the phospholipids, they can mix with water because they're amphipathic. There's parts of them that can mix with water, but lipids consist mostly of hydrocarbon regions. For example, fats, they are very hydrocarbon heavy, and if we know anything about hydrocarbons, a CNA and an H is very nonpolar, and so lipids consist mostly of these hydrocarbon regions, and so that's why they can't, interact with the water around us. If you put a very fatty substance into water, you would see that it would not mix very well at all. It would be very clunky and very clumpy almost, and that's the most important lipids are fats, phospholipids, and steroids, and it's kind of a unifying feature about all of them is that they are not very hydrophilic. We can talk about fats first. Fats are constructed from two types of smaller molecules, glycerol and fatty acids. So we have this glycerol molecule here, and then we have a fatty acid, in this case palmitic acid. And glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon. So we see a hydroxyl group attached to each carbon of this glycerol molecule. And if you look at this graphic already, you can see what's happening here, right? We refer to this as a dehydration reaction. So you can kind of see what's going to happen here. Um, and essentially, a fatty acid consists of a carbonyl group attached to a long carbon skeleton. But at the end of it, there is a hydroxyl group, which is its main goal is to essentially attach to a glycerol to create a part of a triglyceride. And the fatty acids in a fat can be all the same or of 2 or 3 different kinds. For example, we can have palmitic acid here as a fatty acid, but the next two can be different types of fatty acids. And in a fat, those 3 fatty acids join together to the 3 hydroxyl regions of the glycerol here to create what we refer to as a triglyceride. And I refer to as the linkage between two monosaccharides as a glycosidic linkage, but in this case, the linkage between a glycerol and a fatty acid is referred to as an ester linkage. And we see that 3 times, and 3 ester linkages to make up one full triglyceride. And that's essentially what makes up our fats. So I want to go a little bit more into fatty acids. So dehydration synthesis allows the fatty acid to attach to the glycerol, and if this happens 3 times, we have a fat molecule, or in this example, triglyceride, which is the formal name of saying it. They vary in length, the number of carbons, and in the number of locations of double bonds, which is very important. Saturated fatty acids, for example, have the maximum number, or excuse me, not saturated fatty acids. Um, Saturated fatty acids essentially have the maximum number of hydrogen atoms possible and no double bonds. The word saturated means that every carbon is busy, right? If we were to look at the, purest form of a carbon structure, it's when all 4 of its bonds are bonded to something. Um, that's when we have the, best possible scenario for carbon. And essentially what that means is the saturated part comes with the fact that each carbon is essentially bonded to all of its carbons, or all of its carbon bonds. Its 4 bonds are bonded to a hydrogen. Unsaturated fatty acids, on the other hand, are the situation where not all of those carbons are bonded to a hydrogen. There's some bonds that are double bonded, and that's because they're not all busy, and that creates this sort of bend, or this bend structure, which is, a factor of why unsaturated fats, if we were to look at a saturated fat, right, we have this slab of butter, it's solid pretty much, and that's because all of its carbon bonds, all 4 of its bonds, the carbon, are essentially bonded to a hydrogen, and it gets rid of this sort of this bend, or this double bond, which creates this bend in the structure and makes it less sturdier. Whereas in an unsaturated fat, we see that these carbons, not all of them have all 4 bonds which are being used up. Not all 4 bonds are being busy. And so we see this double bond, which causes this bending in the structure, and that's what makes unsaturated fats more more liquidy and less, stronger and sturdier in structure. If we were to look at olive oil, for example, which is a big, concept of unsaturated fats, the reason it's unsaturated is because there's this double bond. It's not all completely bonded, and we have this bend, which, reduces or makes the structure more, less sturdier and less solid. Phospholipids, I have a very, I guess, detailed video dedicated to this, all about the cellular membrane and phospholipids, but this is kind of just a introduction to it. So, in a phospholipid, 2 fatty acids and a phosphate group are attached to a glycerol. The 2 fatty acid tails are hydrophobic, but the phosphate group and its attachments form a hydrophobic head. This is supposed to be a hydrophilic head. Apologies. But this is the hydrophilic head, and this is the hydrophobic tail. And when those phospholipids are added to water, they self-assemble into double-layered sheets called bilayers. Right, we have I think I'll talk about that more in the video, but essentially, if we were to look at the phospholipid bilayer, we have we have the hydrophilic head, and then we have the hydrophobic tails. But underneath that, we have another set of hydrophobic tails and hydrophilic heads, and this is an example of the bilayer. We have um, it's protecting us from the inside of the cell and also from the outside of the cell, so it's selectively permeable. Phospholipids can be dealt in water because they have a charge. They're amphipathic. If we were to look at the phosphate group on the on the top of the head here, right? We have a choline, we have a phosphate, and then we have the glycerol and the 2 fatty acids. These are polar. The phosphate and the choline, or phosphocholine group, are polar, and that's kind of what gives it that ability to interact with water and make this hydrophilic head essentially interacting with water, allows it to interact with water. And then the hydrophobic tails are actually, not all the bonds are essentially double bonded, so they're kind of unsaturated in a way. And these kinks are very important because when we discuss, the emphasis on the fluidity of the cellular membrane, these kinks do come into play because we don't want a cellular membrane to be this sturdy, rigid structure, right? We want it to be flowing to essentially work with the fluidity of the cell, which we'll talk about later. But essentially, the phospholipid bilayer forms a boundary between the cell and its exterior environment. And the hydrophilic head is what allows the phospholipid to work both out and inside the cell, right? It's able to interact with the water, and the tails are able to interact with the hydrophobic parts of the body, like fats. Steroids, I'm going to be very brief about this because this is more important when we talk about the phospholipid bilayer, but they're lipids characterized by a carbon skeleton consisting of four fused rings. Cholesterol, which is a type of steroid, is a component in our cellular membrane and a precursor from which other steroids are synthesized. Um, Testosterone and estrogen, for example, start from the molecule of cholesterol. And we all have some testosterone and some estrogen. You know, some people think that guys only have testosterone, but that's not true. We have little bits of estrogen. It's really just dependent on how much of each is expressed. For example, if you're a male, you can have more testosterone expressed than estrogen, but if you're a female, it's going to be the opposite. And a high level of cholesterol in the blood may contribute to cardiovascular disease. And now we're going to get into probably the most important macromolecule, in my opinion, proteins. Proteins are very, very important, and an example of this is they account for more than 50% of the dry mass of most cells. So if you were to take out all the water from a cell, most of it would just be protein. So enzymes are proteins, for example, and they act as catalysts to speed up a bunch of chemical reactions. They can perform their functions repeatedly, functioning as workhorses that carry out the process of life. Defensive proteins, for example, are antibodies. They're proteins as well that destroy viruses and bacteria. Hormones, which are messengers around the cell that tell the body what's happening. For example, if I were to go in and out, insulin would essentially tell the body to take up all that glucose and, bring our blood sugar back down to normal. Um, Defense, storage, transport, motor proteins, structural proteins, receptor proteins. Proteins are everywhere, and they are the most important, in my opinion, macromolecule because they are essentially the fundamental building blocks of so many of our processes in our body and are very, very important. So that's why having a diet with high protein is important because you're essentially fueling a lot of these important functions that occur in our body. Amino acids, which are essentially the monomers of these proteins, they're all constructed from the same set of 20 amino acids. So we have sets of non-polar amino acids, we have sets of polar side chain amino acids, and we have even pH-based, acidic and basic type amino acid side chains or R groups. The R group is always facing the left. The amino acids differ in the properties due to differing side chains called R groups. So the amino acids generally have the same structure, but the one thing that sets them apart from each is their side chain, their R group, right? For example, glycine has an H as its R group, but if we were to look at something like alanine, it would have CH3 as its R group. So each of these amino acids all have a different type of essentially a different type of R group, which sets them apart from one another. And that R group is very important because the R group tells us what the amino acid is, going to be involved with, right? For example, the nonpolar side chains of this amino acid could be important with, if we were to talk about transport proteins within our phospholipid bilayer, which I'll talk about in another video. But if we were to talk about, how those, if we were to talk about how those transport proteins are probably going to be, hydrophobic on the outside so that they can work with the hydrophobic layer of the phospholipid bilayer, we might see these, non-polar amino acids be on the outside of a transport protein, if that's, one example of it. And then these hydrophilic, the hydrophilic amino acids could be on the inside of a transport protein because those are usually going to be taking in polar things into the body. So they're probably going to want a polar inside of the transport membrane or transport protein. So we might see these hydrophilic um, amino acids be on the inside of a transport protein. They can also deal with acids and bases depending on their environment, similar to how phospholipids can deal with water and without water. Amino acids can deal with a more acidic environment, although that gets into stuff like denaturation, but they can deal with acidic environments and both basic environments. And the bond between amino acids is referred to as a peptide bond. We looked at glycosidic linkages with glucoses, ester linkages with lipids, um, And peptide bonds are essentially what make up amino acids. And a giant structure of amino acids is referred to as a polypeptide, which is essentially a protein. All right, protein structure, which is an important idea when we get into proteins, right? We're looking at the protein as a whole, and so we refer to as these structures sort of, you know, leveled out into the primary, the secondary, the tertiary, and the quaternary structure. The specific activities of proteins result from their intricate three-dimensional architecture. So a functional protein consists of one or more polypeptides precisely twisted, folded, and coiled into a unique shape. So each protein has a generally unique shape. The sequence of amino acids determines a protein's three-dimensional structure. So, what types of amino acids or, its amino acid chain or just essentially the linearity of what type of amino acids are there essentially determine how the um, how the structure will look three-dimensionally. A protein structure determines how it works. The function of a protein usually depends on its ability to recognize and bind to some other molecules. And the primary structure of a protein is its unique sequence of amino acids attached with the peptide bond. So the primary structure is probably the easiest one because it's essentially just, the line of amino acids, the polypeptides. It's just the polypeptide structures as a whole, what the amino acid sequence is. So if I were to just ask you what is the primary structure of this protein, you would just tell me the amino acid sequence of this protein. That's all you would do. You wouldn't tell me anything else because that's just all it is. It's just a linear sequence of amino acids. The secondary structure, which is found in most proteins, consists of coils and folds in the polypeptide chain. So there's two types of them. We have alpha helices and we have beta sheets. And those are just the interactions between the backbones of amino acids. So lots of hydrogen bonding occurs during the secondary structure because that's just sort of the beginning of the folding. And we have two types of them. We have alpha helices and we have beta sheets. Tertiary structures determined by interactions among various side chains, for example, the R groups. If we were to look back here, we could see that these R groups could bind to one another. For example, these non-polar R groups would probably be more clustered together, whereas the polar R groups could hydrogen bond with one another potentially or form bonds. So those R group interactions make up what we call the tertiary structure. The quaternary structure is when a protein consists of multiple polypeptide chains, so just multiple of those polypeptide chains just joining up together and up together to create one big quaternary structure. So quaternary and primary, I think, are probably the easiest ones because quaternary is just a bunch of polypeptides, and the primary structure is just the sequence of amino acids. So those are probably the two easiest ones to remember out of these structures. We get into the secondary structure now. So the coils and folds of secondary structure result from hydrogen bonds between repeating constituents of the polypeptide backbone. So the interactions resulting with hydrogen bonding between the nitrogen and the hydrogen in the amine group. So if we were to look at the amino acids polypeptide backbone, we would see this amine group here, and then essentially that amine group would essentially attach or hydrogen bond with the nitrogen in its amine group, right? Because these can basically hydrogen bond with one another, and if you were to connect all these amino acids together, those hydrogens and those nitrogens would essentially hydrogen bond with one another to sort of create this alpha helix structure or beta pleated sheet. It really just depends on on what type of protein it's going to be coding for, but those essentially connect with one another to create what we refer to as an alpha helix, which can also be turned into a beta pleated sheet. So typical secondary structures are a coil called an alpha helix and a folded structure called a beta pleated sheet. So these are the two types. If you were to be asked a question on like what are the two types of secondary structures in a protein, you could say an alpha helix and a beta pleated sheet, and the those essentially work based on the hydrogen bonding between the backbones of the polypeptide. So tertiary structures, which are the overall shape of a polypeptide, result from interactions between R groups rather than interactions between backbone constituents. So, right, we were talking about the amine group with secondary structures, but tertiary structures bond with R groups and the side chain. So, if we were to look at this graphic here, we could see this essentially, right, we have an R group on an amino acid here, and then on this part of the tertiary structure, we'd have an R group here. And so we'd have these hydrophobic interactions or van der Waals interactions. And essentially what that is they're basically clumping up to one another, right? They're not going to specifically bond together, but they're going to essentially any hydrophobic thing, if it sees another hydrophobic thing, it's going to have hydrophobic interactions or very small type IMFs. It's just going to join together or clump together. And so we'll see that here with these two nonpolar side chains, they're going to want to sort of stick together and clump together. And then for this polar side chain and this other polar side chain, we'll see a hydrogen bond between these two side chains, right? We have an OH and then we have an O that can make a hydrogen bond, so they're going to want to bond together. And then disulfide bridges, which is one of the cooler ones, is essentially if there's a sulfur on the end of one side chain and then the sulfur on the end of another side chain, they're going to create what we refer to as a disulfide bridge, and they're just going to connect to each other. And then we have even ionic bonds between, oops, between acidic and negatively charged side chains and positively charged side chains. Those can create what we refer to as ionic bonds. So there's just a bunch of interactions that are essentially occurring with the tertiary structure between R groups. We see these R groups essentially combining together and forming these bonds. Van der Waals interactions, disulfide bridges, hydrogen bonds, ionic bonds, and these essentially help create the tertiary structure because it's just, the overall folding structure of the protein occurs based off of these R groups interacting with one another. Quaternary structures, they result when two, oh, also want to bring up an important thing here is um, these folding of the hydrophobic or the nonpolar side chains, they usually occur on the inside of a tertiary structure, right? Because if we were to look at a protein and it's in, an aqueous environment, right? Our body is very aqueous. The protein is going to want to make sure that the fats, or not the fats, but the non-polar side chains are located inside of the protein so they don't interact with the water which is on the outside. And so what we'll see here is with the folding structure of a tertiary structure is that these hydrophobic molecules, or excuse me, these hydrophobic side chains, they're going to be tucked inside of the protein or the tertiary structure of the protein so that they have as little interaction with water as possible, and they're just going to be bunched up together. And so the polar groups, or the ones that can interact with water, are usually going to be on the outside because they can interact with an aqueous environment so that there's more space on the inside of a tertiary structure to allow those non-polar side chains to be clumped up together and stay away from the water as much as possible. So that's another very, very cool thing about biology and how it's able to sort of work within its environment. Quaternary structure results when two or more polypeptide chains form one macromolecule. So collagen is a fibrous protein consisting of three polypeptides coiled together like a rope. Hemoglobin, which is a very important quaternary structure for oxygen transport in our body and allows our tissues to be oxygenated, to have oxygenated blood, is a globular protein consisting of four polypeptides, two alpha and two beta subunits. So obviously we have the heme iron here where the oxygens are going to bind to these iron groups and essentially allow this, hemoglobin molecule to transport oxygen to tissues that need it. So these are just examples of quaternary structures, and these are very, very, very big proteins that are just multiple polypeptide chains linked together upon one another. So we have hemoglobins, for example, and then we have collagen, which is another big one. We're done with proteins now. I guess we can start moving on to nucleic acids, which a very important thing that I want to talk about is the central dogma, which is, the translation of protein, right? The proteins are very important for our processes in life, and they express a lot of things, right? The proteins express, our phenotypic natures, right? So our DNA is essentially going to turn into RNA through a process referred to as transcription, and that RNA is going to be translated into protein. So the synthesis of mRNA will go through, its specific, you know, bi-prime caps, all that kind of stuff, pre-mRNA processing, moving into the cytoplasm, and it'll be synthesized by ribosome into a long polypeptide chain. So the amino acid sequence of a polypeptide is programmed by a unit of inheritance called a gene. So if you wanted to know how these, proteins are made, they're made through a process through central dogma, right? The GNA or gene expression kind of tells us what the amino acid sequence will be so that it could be translated into a specific protein that will essentially enact those changes and essentially show those changes. So genes consist of DNA and nucleic acid made of monomers called nucleotides. There's two types of them we refer to as DNA and RNA. So deoxyribonucleic acid and ribonucleic acid. DNA provides directions for its own replication, so basically tells itself what to replicate into, which we'll get into when we talked about DNA replication with our mitosis and meiosis video. And DNA directs synthesis of messenger mRNA or messenger RNA or mRNA. And through mRNA, it controls protein synthesis, so we want those proteins to be made. Well, DNA is essentially going to tell not the body, but it's essentially going to express those genes, and those genes are going to create mRNA, which is then going to be translated into protein. And then, just to go through each of these bullet points, this process is called gene expression. So each of our cells essentially has the same identical genomic DNA or the same sets of genes, but how they're expressed is actually what determines, how we look like, right? Certain genes are turned off, certain genes are turned on, and the DNA is essentially going to express those genes through proteins. And so the expressions of our genes essentially tell us how we'll look, right? The color of our eyes is a set of proteins that are expressed by the DNA, and it's a very big process we'll get into talk about gene expression. Nucleic acids continued. So nucleic acids are essentially polymers called polynucleotides. So you can refer to them as nucleic acids or you can refer to them as polynucleotides. And each polynucleotide is made of monomers called nucleotides. So important thing I want to talk about is nucleotide is the nucleoside plus the phosphate group. And the nucleoside is the nitrogenous base and the sugar. So we'd have the nitrogenous base and the sugar here, which makes up a nucleoside. And a nucleotide is that plus the phosphate group. So each nucleotide consists of a nitrogenous base, a pentose sugar, and one or more phosphate groups. Pentose meaning a 5 prime sugar or 5 carbon sugar. There are two families of nitrogenous bases. We have pyrimidines, which are cytosine, thymine, and uracil because they have a single 6-membered ring. So they have, essentially a 6-member ring right here. We have 1, 2, 3, 4, 5, 6. And then purines, on the other hand, which are just adenine and guanine, have a 6-member ring fused to a 5-member ring. And in DNA sugar, deoxyribose, we actually don't have that OH, right? Deoxy means that we lose that oxygen, whereas in ribose, we still have that oxygen. So the biggest difference between the two pentyl sugars of deoxyribose, of DNA and RNA is deoxyribose is in the name, we're getting rid of that oxygen, we're losing this oxygen, whereas ribose, we still have that oxygen attached to it. So those are just some important, things to remember when you're looking at specifically the type of DNA, right? If you look at the pentose sugar, it tells you a lot about if you're looking at DNA or RNA. So DNA molecules have two polynucleotides spiraling around an imaginary axis, forming a double helix, and the backbones run in opposite directions from 5' to 3'. So they're essentially anti-parallel, and one DNA molecule includes many genes. Only certain bases in DNA pair up and form hydrogen bonds. For example, adenine always pairs up with thymine, and guanine always pairs up with cytosine, and this is called complementary base pairing. This feature of DNA structure makes it possible to generate two identical copies of each DNA molecule in a cell preparing to divide. We talked about this in the mitosis and meiosis video. When we go through DNA synthesis, we're going to need to make two sister chromatids, right? And that is a process of DNA replication, which occurs with the complementary base pairing. It just really helps a lot with creating two identical copies. RNA, in contrast, is single-stranded. DNA is double-stranded. RNA is single-stranded. Complementary pairing can also occur between two RNA molecules or between two parts of the same molecule. In an RNA, thymine is replaced by uracil, so A and U pair while DNA exists as a double helix. RNA molecules are more variable in form. And I want to go back here and to kind of just talk about how each of these bonds are formed, right? We were talking about proteins and their peptide bonds and dehydration synthesis. But in here, essentially, right, we have these nitrogenous bases, and they are going to bond together to essentially create the DNA backbone. How does that all work? So, if we were to look at the structure of, for example, DNA, right? If we were to look at this 1, 2, 3 prime end of this sugar, this 3 prime end is going to essentially OH, and we have this O at the bottom of phosphate group, H2O. Essentially what's going to happen is this OH, this hydroxyl group and this oxygen are going to create H2O, and that's going to be removed to create this linkage between this pentose sugar and this phosphate group. And that'll essentially happen here. This phosphate group will link with the 5 prime end of this pentose sugar, and essentially keep doing that same thing over and over and over again to create the sugar phosphate backbone, which is very important for the structure of the DNA. And the same situation happens here, right? If we were to have, for example, adenine, which is a purine, it's going to want to bind to thymine. The same thing is going to happen here. This OH structure and this hydroxyl structure will essentially go through a process of dehydration synthesis to attach adenine and thymine together, and that's essentially how those nitrogenous bases are attached. So polypeptide backbone, dehydration synthesis, the two nucleotides, the two complementary nucleotides attaching together, also dehydration synthesis. Dehydration synthesis is very important for so many processes in our body, especially for the formation of DNA as a whole, nucleic acids as a whole. Um, DNA or RNA, we need dehydration synthesis. It's another example of why water is life, because it powers so many processes in our body. And so those will essentially create the sugar phosphate backbone, and then we'll see another linkage between this nitrogenous base and its other nitrogenous base to create this structure of DNA. And that is our essential video on macromolecules. I hope you guys enjoyed the video, and I'll be posting a lot more frequently. Um, just been very busy recently, so I'll be trying to get back into the habit of posting. But thank you guys for watching, and I'll see you next time.